Hey BioGeeks, we are going to look at one of the stop and think slides in your notes on blood. And this one is on blood transfusion. So before you tackle this kind of problem, you have to understand the difference between an antigen and an antibody. So antigens are like name tags on cell membranes. They identify the cells. And because your immune system doesn't want foreign substances or foreign cells or anything in your body. So when we get a bacterial infection or a viral infection or even an organ transplant or the wrong kind of blood type in our body, our immune system recognizes foreign antigens, which means antigens that are different from anything in our own body. And if they're foreign, our immune system will attack. And we'll look at that in a few minutes. If they're not foreign, then everything's okay, which is why we need to be careful when we are giving blood to patients. If we give the wrong kind, bad things can happen. Something called agglutination, which I'll show you in a few minutes. All right, let's look at this problem. So Lauren is blood type A, which means she's going to have A antigens. I'll show you a little drawing of that in a second. And she's going to receive a transfusion with blood type AB. We want to know, is this safe? And then I want you to draw a sample of Lauren's blood interacting with the donor's blood. And RBCs, okay, RBCs does not stand for Royal Bank of Canada. It stands for red blood cells. We can also call those erythrocytes. And that's the type of cell we're looking at with regards to antigens and determining whether a blood transfusion is safe or not. It's a whole lot more complicated than that, but that's the main blood cell we are concerned about with regards to transfusions. All right, let's have a look at that on the paper. All right, I've got things started here. Oops. And so Lauren is type A, and Lauren is the recipient, which means she's going to receive the blood transfusion. And her red blood cells, because she is type A, are going to have only the A antigen. All right. So if we look at the blood types in the ABO blood group, we have type A, B, AB, and O. And we can also look at what antigens those types have. Type A will have A antigens. Blood type B will have B antigens. Blood type AB will have both A and B. Now, be careful. A lot of students will think there's such thing as a AB antigen. Not true. If you're blood type AB, it means you have both A and you have B. If you're type O, think of O as 0. You don't have any or either of these antigens. So we don't have them. There's not an antigen O, not a thing. And antibodies, excuse my messy writing, if you're blood type A, what would be the foreign antibodies? Well, the only other one is B. So if your blood type is A, you will have anti-B antibodies. If your blood type is B, you'll have anti-A antibodies. If your blood type is AB, that means you have both the A and the B antigens, so neither are foreign, so you won't have any antibodies to attack either of those. If your blood type is O, you don't have either of those antigens, so you're going to have both anti-A and anti-B antibodies. Okay, let's get into our problem. So Lauren is type A, so she has A antigens, and our donor is AB, so the donor has both A antigens and B antigens. Now, let's do this. So we're going to have type AB, the donor, is going to be going into Lauren's body, who is type A. The best way I find to get your head around this is put yourself in Lauren's shoes. And her immune system is going to say, is there anything foreign coming into my body? Or in other words, are there any foreign antigens coming into my body? So with this blood coming into Lauren's body, A is not foreign because Lauren has A antigens. However, we have a problem. B is foreign. So this is what's going to cause a problem. And remember, if Lauren is type A, she is going to have anti-B antibodies. And antibodies are Y-shaped proteins. I'll just put a little B there just to remind us that it's the B antigens that she will attack, okay? And these antibodies actually are shaped like letter Ys, and this area right here and here has a special configuration where it can actually physically attach to B antigens. And somebody with anti-A antibodies would have antibodies with different configurations on the tips here that would attack the A antigens. So when we get a bunch of these donor red blood cells coming into Lauren's body, let's draw a few of those. So remember, it's type AB, so the donor is going to have both A and B antigens. All right, so when you do this, I want you to draw a number of these donor cells. Okay, so remember, Lauren is type A, so she does not have any issue with the A antigens coming into her body, but because she does not have any B antigens, those Bs are foreign to her body. Okay, so her anti-B antibodies are actually going to attack these parts of the donor cell, and they get mobilized like this. So here's an antibody, and I'm going to draw a few more donor cells in here. There is an anti-B antibody attacking this B antigen. It physically attaches. And I'll just draw another donor cell there. So it can attach this one, and it kind of holds them together like that. All right, and there's a special word for that I'll mention in a second here. And we're not going to attack the A antigens because those are not foreign. We don't really recognize those. All right, there's another anti-B antibody attacking those B antigens. And we have it going on here as well. Boom, boom. I'm not an artist, so bear with my messy sketching. So what we have here is something called clumping. Now, this is not clotting. That is an entirely different process. This is clumping. They get clumped together. So think of the antibodies like Velcro that's kind of holding them together. And there's another process that happens in terms of what's happening down the road here and why this causes a problem we won't get into. Now, a fancy word for clumping, which I do want you to know, is agglutination. Agglutination and clumping, all the same thing. And so one thing that happens after this is these red blood cells are basically marked for death, and there's other sequence coming along that's going to cause the red blood cells to burst open, and the hemoglobin, which is the protein in the red blood cells that actually carries oxygen, that's going to kind of leak out into the plasma or fluid part of the blood, and outside the red blood cells it's toxic. That's going to cause some problems. Also, when you have this clumping together or agglutination of red blood cells, you can imagine that as we are, oops, I'm off screen here, when we have a clump of red blood cells moving through a small blood vessel, as that gets farther and farther away from the heart, it branches out, and the blood vessels get smaller, so it physically can't go there. So it kind of blocks the passage of blood. And the cells that are being supplied by oxygen by these blood vessels, they're, let's say it's a muscle cell, nucleus, muscle cell. If blood can't flow through here, they get deprived of oxygen, and we have the death of tissue. All right, so quick little recap. We have Lauren is the recipient. She's going to receive the transfusion. She's type A, which means she has A antigens. And in terms of this ABO blood group, we're not talking about antibodies for measles and smallpox and the common cold and so on. We're only talking about the antibodies that will be produced and mobilized in blood transfusions. So with regard to blood transfusions, she can't have any blood type that has a B antigen because for her, Bs are foreign. All right. The donor is AB. Oh, that's a problem because the B has foreign antigens. So this is not a safe transfusion. Lauren has anti-B antibodies. They are going to attach to the donor's cells. Specifically, when you draw this, don't just draw the antibody randomly attached anywhere. I want you to draw it on the actual B antigens. That's where they're going to attach. And because this causes the red blood cells to clump up, and that's called agglutination, right? And later on, we won't learn about this process, but the red blood cells burst open. The hemoglobin leaks out into the tissues or into the blood plasma, and that is toxic to tissues. And we also block the passage of this clump of cells getting through smaller blood vessels. So there is an example of an unsafe blood transfusion.